Hypersensitivity. Hypersensitivity is a term referring to an excessive and undesirable reaction produced by the normal immune system in response to a stimulus. All types of hypersensitivity reactions require a pre-sensitized host. This means an individual who has been previously exposed to an antigen and who has successfully mounted an immune response to that antigen. At the time of the second exposure, the excessive reaction may result in unpleasant side effects. Tissue injury in these type of reactions is mediated by the immune system of the body and may be caused by either humoral or cell-mediated immune mechanisms. Depending on the precise immunological mechanisms involved, hypersensitivity is classified into 4 types. These are type 1 or immediate type, type 2 or antibody-mediated type, type 3 which is immune complex mediated, and type 4 which is cell-mediated hypersensitivity. Let us look at these a little more closely. Type 1 hypersensitivity is a rapid immunological reaction which occurs within minutes after exposure to the antigen. That is, the combination of the challenging antigen with preformed antibody which is found usually bound to the surface of mast cells in individuals who have been previously sensitized. This type of reaction is also called an allergy, and the antigens that evoke this response are called allergens. These allergens may reach the body through the food as airborne particles. They may be viruses. The manifestations of type 1 hypersensitivity are of 2 types. They may be either systemic, in which there is a state of shock within minutes, and this at times may be fatal, or it may manifest as local reactions. These local reactions include acute urticaria on the skin in response to various stimuli. This may be allergic rhinitis involving the nasal mucosa caused by pollen or dust mite feces. Asthma is a counterpart of allergic rhinitis involving the bronchial mucosa, and food allergy may be seen involving the gastrointestinal mucosa. When the challenging antigen or allergen, of which penicillin is a classic example, first enters the immune system of the body, it is picked up by the dendritic cells and presented to naive CD4 positive helper T cells. These naive cells are thus stimulated to differentiate into Th2 cells. These cells secrete cytokines IL-5, 4 and 13. IL-4 acts on B cells and switches them into IgE producing plasma cells. Also promotes formation of more Th2 cells. The IgE antibody molecules bind to mast cells. IL-5 activates eosinophils and IL-13 further promotes IgE production. After this stage, when the immune system has been primed and a state of hypersensitivity is present, a second challenge by the same allergen leads to extensive degranulation of the mast cells. This releases large number of primary and secondary mediators and causes the various effects of the allergic reaction, systemic or local.